Tonight's lecture is over Chapter 4, Cellular Structure. Each cell in the human body is a miniature marvel of great complexity. If a machine with millions of parts, say a jumbo jet, were reduced to microscopic size, its complexity would not begin to rival that of a living cell. Organisms are either single-celled, such as most prokaryotes and protists, or multicelled, such as plants, animals, and most fungi. Your own body is a cooperative society of trillions of cells of many specialized types. 3 examples are muscle cells that keep your heart beating, the nerve cells that control your muscles, and the red blood cells that carry oxygen throughout your body. Everything you do, every action, every thought reflects processes that occur at the cellular level. And for understanding the world of cells, one of our main tools is the microscope. Our understanding of nature often parallels the invention and refinement of instruments that extend the human senses. For example, before microscopes were first used in the 1600s, no one knew that living organisms were composed of cells. The type of microscope used by Renaissance scientists is the same kind of microscope you will use in the biology lab. This is called a light microscope. Visible light is projected through the specimen, such as a single-celled protist. Glass lenses enlarge the image and project it into a human eye or a camera. 2 important factors in microscopy are magnification and resolving power. Magnification is an increase in the object's apparent size compared with its actual size. The clarity of that magnified image depends on resolving power, that is, the ability of an optical instrument to show 2 objects as separate. For example, what appears to the unaided eye as 1 star may be resolved as 2 stars with a telescope. Each optical instrument, be it an eye, a telescope, or a microscope, has a limit to its resolving power. The human eye can resolve points as close together as 0.1 millimeter. That's about the size of a very fine grain of sand. However, for light microscopes, the resolving power is about 0.2 micrometers. That's the size of a small bacterial cell. This limits the useful magnification to about 1,000 times. With greater magnification, the image becomes blurry. Cells were first described in 1665 by the British scientist Robert Hooke. Hooke used a microscope to examine a thin slice of cork from the bark of an oak tree. For the next 2 centuries, scientists found cells in every organism that was examined with the microscope. By the mid-1800s, this accumulation of evidence led to the cell theory, which states that all living things are composed of cells and that all cells come from other cells. Then in the 1950s, our knowledge of cell structure took a giant leap forward with the invention of electron microscopes. Instead of using light, an electron microscope uses a beam of electrons to resolve images. It has much better resolving power than the light microscope. So really there are 4 types of microscopes that you should be familiar with at this point. The light microscope, the dissecting microscope or the stereo microscope, and the scanning electron microscope, and the transmission electron microscope. So S for scanning electron microscope and stereo microscope. The stereo microscope and the scanning electron microscope are analogous in the respect that they allow the viewing of the surface of an object. That means that either the electrons or the light is projected onto the surface of a specimen. On the other hand, the transmission electron microscope and the light microscope are analogous in the respect that they allow electrons or light to be projected through a specimen. Thereby, the observer can view the inside, the internal structure of a specimen. And this figure on this slide just shows different sizes of specimens, and it shows the resolution of the various microscopes that we've been talking about in contrast to the naked eye. Moving on to the next slide, all cells share two common features, and that is they have a plasma membrane. That's the outer layer that separates the cell's interior from its surrounding environment. And they have cytoplasm, which is the water-based center of the cell. The cellular structures, if there are any, are found in the cytoplasm. So yes, a cell has to have a plasma membrane, otherwise it's not a cell. And remember from our discussion on the macromolecules that the plasma membrane of a cell is composed of the phospholipid bilayer, which has a hydrophilic head and a hydrophobic tail. So when that molecule comes in contact with water, the phospholipid bilayer orients itself and creates a plasma membrane, and that is how a cell is formed. So on this next slide, we're going to start talking about the two major categories of cells, and we'll start with prokaryotic cells. The countless cells that exist on the earth fall into two basic categories, prokaryotic cells and eukaryotic cells. Prokaryotic cells characterize the organisms of the domains bacteria and archaea. These are collectively known as prokaryotes. Organisms of the domain eukarya, protists, plants, fungi, and animals, are composed of eukaryotic cells and are called eukaryotes. So as this cell outlines, prokaryotic organisms are in general simple, single-celled or unicellular organisms. Their DNA is loose in the cytoplasm. They have ribosomes because ribosomes are needed to make proteins. Some prokaryotic organisms will have a cell wall, which will cover the plasma membrane. And some prokaryotic organisms have a capsule that can cover the cell wall. Others may have flagella, pili, or fimbriae. And the cartoon on this slide of a prokaryotic cell just shows a generalized view of a prokaryotic cell. Now bear in mind that not all prokaryotic cells are going to look like this. Not all prokaryotic cells will have all these features. The main gist of a prokaryotic cell is that it's going to fall into the domain bacteria or archaea, and it's likely a single-celled organism with no membrane-bound organelles. Also, another important distinction of a prokaryotic cell from a eukaryotic cell is that prokaryotic cells are older in an evolutionary sense. The fossil record indicates that the first prokaryotes appeared on the planet Earth about 3.5 billion years ago, whereas the first eukaryotes did not appear until about 2.1 billion years ago. Also, prokaryotic cells are in general smaller than eukaryotic cells, and they also have a much simpler structure than the eukaryotic cell does. As I've already mentioned, the eukaryotic cell is going to have the membrane-bound organelles, which the prokaryotic cell is going to lack. Consider this analogy. A eukaryotic cell is like an office that is separated into cubicles. With each cubicle, a specific function is performed, thus dividing the labor among many internal compartments. The cubicle boundaries within the eukaryotic cells are made from membranes that help maintain a unique chemical environment inside of each cubicle. In contrast, the interior of a prokaryotic cell is like an open warehouse. The spaces used for specific tasks are distinct, but not separated by barriers. So let's refer back to our cartoon on this slide. So surrounding the plasma membrane of most prokaryotic cells is a rigid cell wall which protects the cell and helps it to maintain its shape. In addition, some prokaryotes have another sticky outer layer called a capsule, and this surrounds the cell wall. Capsules provide protection and help prokaryotes to stick to some surfaces. Some prokaryotes have short projections called pili, which can also attach to surfaces. Many prokaryotic cells have flagella, which are long projections that help them propel through their liquid environment. Moving on to eukaryotic cells, all eukaryotic cells, whether from animal, plants, protists, or fungi, are fundamentally similar. The entire region of the cell between the nucleus and the plasma membrane is called the cytoplasm. This term is also used to refer to the interior of a prokaryotic cell. The cytoplasm of a eukaryotic cell consists of various organelles suspended in fluid. Most membrane-bound organelles of eukaryotic cells are found in both plant and animal cells. But one important difference is the presence of chloroplasts in plant cells, but not in animal cells. Chloroplasts are organelles that convert light energy into the chemical energy of food. Another distinction is that plant cells have a protective cell wall outside the plasma membrane. Eukaryotic cells are in general larger than prokaryotic cells, and remember, prokaryotic organisms are most often single-celled organisms or unicellular. In the eukaryotic domain, however, organisms can be either unicellular or multicellular. Protists, for instance, are examples of single-celled eukaryotic organisms. They are eukaryotic because they have the membrane-bound organelles that distinguish eukaryotic cells from prokaryotic cells. Moving on to the shared characteristics of eukaryotes. Most eukaryotes are going to have a nucleus surrounded by a nuclear envelope with nuclear pores. Most will have mitochondria. Most will have a cytoskeleton containing the structural and motility components of the cell. Some eukaryotic cells will have flagella and cilia, and these are also organelles associated with cell motility. Many eukaryotic cells have chromosomes. Mitosis is a process of nuclear division, which we will learn about in subsequent chapters. But that's how cells replicate themselves, is through mitosis. Unless it's a sex cell, then the process is called meiosis. Also, many eukaryotes are sexually reproducing organisms, and many eukaryotes are going to have cell walls. Remember, this is just some shared characteristics of eukaryotes. Not every eukaryote has all of these features. Moving on to endosymbiosis and the evolution of eukaryotes. Symbiosis is the close association between organisms of two or more species, and endosymbiosis refers to one species living inside of another host species. Chloroplasts and mitochondria seem to have evolved from small symbiotic prokaryotes that establish residence within other larger host prokaryotes. The ancestors of mitochondria may have been aerobic bacteria that were able to use oxygen to release large amounts of energy from organic molecules through a process called cellular respiration. At some point, such a prokaryote might have been an internal parasite of a larger heterotroph, or an ancestral host cell may have ingested some of these aerobic cells for food. If some of the smaller cells were indigestible, they might have remained alive and continued to perform respiration in the host cell. In a similar way, photosynthetic bacteria ancestral to chloroplasts may have come to live inside of a larger host cell. Some scientists argue that since most eukaryote cells have mitochondria, but only some have chloroplasts, that mitochondria evolved prior to chloroplasts. Anyway, however the relationship began, it is not hard to imagine how a symbiosis between engulfed aerobic or photosynthetic cells and a larger host might have become mutually beneficial. In a world that was becoming increasingly aerobic, a cell that was itself an anaerobe would have benefited from aerobic endosymbionts that turned the oxygen to an advantage. And a heterotrophic host could derive nourishment from photosynthetic endosymbionts. In the process of becoming more interdependent, the host and endosymbionts would have become a single organism and their parts inseparable. And on this next slide, this table details the components of prokaryotic and eukaryotic cells and their functions. So we can see we have the cell component, the function. Is it present in prokaryotes? Is it present in animal cells? Is it present in plant cells? So for instance, the plasma membrane. Its function is to separate the cell from the external environment. It controls the passage of organic molecules, ions, water, oxygen, and waste into and out of the cell. Is this present in prokaryotes? Yes. Is it present in animal cells? Yes. Is it present in plant cells? Yes. The plasma membrane is something that all cells have in common. So I'd like for you to take a few minutes to go through this table on your own time. Moving on to the next slide. This slide is just a continuation of the components of prokaryotic and eukaryotic cells and their functions. So again, please take a few minutes to go through this table on your own time. Moving on to cytoplasm. Cytoplasm is a water-based fluid inside of all cells. Sometimes it's referred to as the cytosol, and it contains ions, small molecules, and macromolecules. Moving on to the nucleus. The nucleus is bordered by a double membrane called the nuclear envelope. Each membrane of the nuclear envelope is similar in structure to the plasma membrane. Pores in the envelope allow certain materials to pass in between the nucleus and the cytoplasm. Within the nucleus, long DNA molecules and associated proteins form fibers called chromatin. Each long chromatin fiber constitutes one chromosome. The number of chromosomes in a cell depends on the species. For example, each human body has 46 chromosomes, whereas rice cells have 24 and dog cells have 78. The nucleolus is the prominent structure within the nucleus. This is the site where the components of ribosomes are made. Moving on, the nucleus connects to the endoplasmic reticulum. Moving on, the endoplasmic reticulum is one of the main manufacturing facilities within a cell. It produces an enormous variety of molecules. Connected to the nuclear envelope, the endoplasmic reticulum forms a labyrinth of tubes and sacs running throughout the cytoplasm. The endoplasmic reticulum separates its internal compartment from the surrounding fluid in the cytoplasm. The endoplasmic reticulum is sometimes referred to as the ER. There are two components that make up the ER, rough ER and smooth ER. The two types of ER are physically connected but differ in structure and function. The rough ER refers to the appearance of this organelle in electron micrographs. The roughness is due to ribosomes that stud the outside of the ER membrane. These ribosomes produce membrane proteins and secretory proteins. Some newly manufactured membrane proteins are embedded right in the rough ER membrane. Thus, one function of the ER is to produce new membrane. Secretory proteins are those that are exported or secreted to the fluid outside of the cell. Cells that secrete a lot of protein, such as the cells in your salivary glands, which secrete enzymes into your mouth, are especially rich in rough ER. Some products manufactured by the rough ER are dispatched to other locations in the cell by means of transport vesicles. These are membranous spheres that bud from the rough ER. On the other hand, the smooth ER refers to the fact that this organelle lacks the ribosomes that populate the surface of the rough ER. A diversity of enzymes built into the smooth ER membrane enables this organelle to perform many functions. One is the synthesis of lipids, including steroids. For example, cells in the ovaries or testes that produce the steroid sex hormones are enriched with smooth ER. In liver cells, Enzymes of the smoothie are detoxify circulating sedatives such as barbiturates, stimulants such as amphetamines, and some antibiotics. As liver cells are exposed to a drug, the amounts of smoothie are and its detoxifying enzymes increase. This can strengthen the body's tolerance of the drug, meaning that higher doses will be required in the future to achieve the desired effect. The growth of smoothie R in response to one drug can also increase tolerance of other drugs. Barbiturate use, for example, may make certain antibiotics less effective by accelerating their breakdown in the liver. Furthermore, increased tolerance of drugs is one of the hallmarks of addiction. Addiction is a potentially serious consequence of the continued use of certain drugs. The Golgi apparatus, an organelle named for its discoverer, Camillo Golgi, is a refinery warehouse and shipping center. Working in close partnership with the ER, the Golgi apparatus receives, refines, stores, and distributes chemical products of the cell. Products made in the ER reach the Golgi in transport vesicles. One side of a Golgi stack serves as a receiving dock for the vesicles. The shipping side of the Golgi stack is a depot from which finished products can be carried in transport vesicles to other organelles or to the plasma membrane. Vesicles that bind with the plasma membrane transfer proteins to it or secrete finished products to the outside of the cell. Products of the ER are usually modified by enzymes during their transit from the receiving to the shipping side of the Golgi. For example, molecular identification tags such as phosphate groups may be added that serve to mark and sort protein molecules into different batches for different destinations. Moving on to ribosomes. Ribosomes are responsible for protein synthesis. In eukaryotic cells, the components of ribosomes are made in the nucleus, then transported through the pores of the nucleus into the cytoplasm. It is in the cytoplasm that the ribosomes begin their work. Some are suspended in the fluid of the cytoplasm, making proteins that remain in the fluid. Other ribosomes are attached to the outside of the endoplasmic reticulum. These ribosomes make proteins that are incorporated into the membranes or secreted by the cell. It's important to note that ribosomes are also found in prokaryotic cells. Moving on to the next slide. Mitochondria are the sites of cellular respiration. This is a process that harvests energy from sugars and other food molecules and converts it to another form of chemical energy called ATP. Cells use molecules of ATP as a direct energy source for most of their work. In contrast to chloroplasts, mitochondria are found in almost all eukaryotic cells, including your own. An envelope of two membranes encloses the mitochondrion, which contains a thick fluid called the matrix. The inner membrane of the envelope has numerous foldings called cristae. Many of the enzymes and other molecules that function in cellular respiration are built into the inner membrane. By increasing the surface area of this membrane, the cristae maximize ATP output. Besides their ability to provide cellular energy, mitochondria and chloroplasts share another unique feature among eukaryotic organelles. That is, they contain DNA. This fact is evidence that supports endosymbiotic theory. Moving on to the cytoskeleton, if someone asks you to describe your house, you would most likely mention the various rooms and their locations. You probably would not think to mention the foundation and beams that support the house. Yet these structures perform an extremely important function. Similarly, cells have an infrastructure called a cytoskeleton. This is a network of fibers extending throughout the cytoplasm. The function of the cytoskeleton is to provide mechanical support for the cell and allow movement. This is very important for animal cells that lack rigid cell walls. The cytoskeleton contains several types of fibers made from different types of protein. One important type of fiber are microtubules. Microtubules are straight, hollow tubes composed of proteins. The other kinds of cytoskeletal fibers, called intermediate filaments and microfilaments, are thinner and solid. Just as the bony skeleton of your body helps fix positions of your organs, the cytoskeleton provides anchorage and reinforcement for many organelles in a cell. For instance, the nucleus is often held in place by a cytoskeleton cage of filaments. Other organelles move along tracks made from microtubules. For example, a lysosome might reach a food vacuole by moving along a microtubule. Microtubules also guide the movement of chromosomes when cells divide in a process called mitosis or meiosis. The cytoskeleton is dynamic. It can quickly dismantle in one part of a cell by removing protein subunits and reform in a new location by reattaching the subunits. Such rearrangement can provide rigidity in a new location, change the shape of the cell, or even cause the whole cell or some of its parts to move. This process contributes to the amoeboid movements of the protist amoeba and some of our white blood cells. Continuing with microtubules, we'll shift gears and talk a little bit about flagellum and cilia. In some eukaryotic cells, a specialized arrangement of microtubules functions in the beating of flagella and cilia. Cilia and flagella are motile appendages, that is, extensions from a cell that aid in movement. Eukaryotic flagella propel the cell by an undulating whip-like motion. They often occur singly, such as in the sperm cells of humans and other animals. Cilia are generally shorter and more numerous than flagella and promote movement by a coordinated back and forth motion, like the rhythmic oars on an 8-person crew team. Both cilia and flagella propel various protists through water. Though different in length, number per cell, and beating pattern, cilia and flagella have the same basic architecture. That is, a core of microtubules wrapped in an extension of the plasma membrane. Some cilia extend from non-moving cells that are part of a tissue layer. There, they move fluid over the tissue surface. For example, the ciliated lining of your windpipe helps cleanse your respiratory system by sweeping mucus with trapped debris out of your lungs. Tobacco smoke can inhibit or destroy these cilia, interfere with normal cleansing mechanisms, allowing more toxin-laden smoke particles to reach the lungs. Frequent coughing, which is common in heavy smokers, then becomes the body's attempt to cleanse the respiratory system. Also, because human spur rely on flagella for movement, it's easy to understand why problems with flagella can lead to male infertility. Sperm with malfunctioning flagella cannot travel up their female reproductive tract to fertilize an egg. Interestingly, some men with a type of hereditary sterility also suffer from respiratory problems. The explanation lies in the similarities between flagella, which is found in sperm, and cilia, which is found in the lining of the respiratory tract. Because of a defect in the structure of their flagella and cilia, their sperm do not swim, and this causes sterility. And, to top it off, their cilia do not sweep mucus out of their lungs, and this is cause for recurrent respiratory infections. Moving on to the plasma membrane. The plasma membrane and other membranes of the cell are composed mostly of lipids and proteins. The lipids belong to a special category called phospholipids. They are related to dietary fats but only have 2 fatty acid tails instead of 3. In place of the third fatty acid, a phospholipid has a phosphate group. The phosphate group is electrically charged, making it hydrophilic. But 2 fatty acid tails are hydrophobic. Thus, phospholipids have a chemical ambivalence in their interactions with water. The phosphate group, or the head, mixes with the water, while the fatty acid tail avoids it. This makes phospholipids good membrane material. By forming a 2-layered membrane, or phospholipid bilayer, The hydrophobic tails of the molecules stay away from water while the hydrophilic heads remain surrounded by water. Embedded in the phospholipid bilayer of most membranes are proteins that help regulate traffic across the membrane and perform other functions. Membranes are not static sheets of molecules locked rigidly in place. The phospholipids and most of the proteins are free to drift about in a plane of the membrane. Thus, a membrane is a fluid mosaic. Fluid because the molecules can move freely past one another, and mosaic because of the diversity of proteins that float like icebergs in the phospholipid sea. Plant cells contain a cell wall which is made of cellulose. The cell wall is a rigid covering that provides structural support and gives the cell shape. Most of the living world runs on energy provided by photosynthesis. Photosynthesis is the conversion of light energy from the sun to chemical energy of sugar and other organic molecules. Chloroplasts, which are unique to the photosynthetic cells of plant and algae, are the organelles that perform photosynthesis. Plasmodesmata are channels running through cell walls. Plasmodesmata connects adjacent plant cells' cytoplasm and allows molecules to travel from plant cell to plant cell. A lysosome is a sac of digestive enzymes found in animal cells. Lysosomes develop from vesicles that bud off from the Golgi apparatus. Enzymes within a lysosome can break down large molecules such as proteins, polysaccharides, fats, and nucleic acids. The lysosome provides a compartment where the cell can digest these molecules safely without unleashing the digestive enzymes into the cell itself. The extracellular matrix is an intricate network composed of an array of multi-domain macromolecules organized in a cell tissue-specific manner. Components of the extracellular matrix link together to form a structurally stable composite, contributing to the mechanical properties of tissues. Intracellular junctions allow cell-to-cell communication. One type of intercellular junction is called a tight junction, and tight junctions connect cells tightly together, so tight that it may be waterproof. In other words, there's nothing that can squeeze between these two cells. Desmosomes are a structure by which two adjacent cells are attached. They are formed from protein plaques in the cell membranes, and they are linked by filaments. Gap junctions are channels between adjacent cells that allow molecules to travel from cell to cell. And this next slide just shows cartoons of the cellular connections we were talking about on the previous slide. So let's recap tonight's lecture. After tonight, you should know what the 3 points of unified cell theory are. You should know when would different types of microscopes be used and what they would be used for. You should be able to list features that are common to all cells. You should know the 3 domains of life: archaea, bacteria, eukaryota. You should know how to identify a eukaryotic cell versus a prokaryotic cell. You should be able to describe endosymbiotic theory. You should be able to identify all of the organelles in a eukaryotic cell. You should be able to match each eukaryotic organelle with its function. Furthermore, you should know what the job of the cytoskeleton is and what events microtubules are involved in. You should also be able to tell the difference between cilia and flagellum. You should also know what organelles are unique to animals and unique to plants. And you should also know how plant cells and animal cells are connected to each other.